Uh, Welcome, everyone, and thank you for joining us from all over the world. Just a quick note, today's presentation will be recorded and available for those who could not attend. My name is Sabrina Merchant. I have a 10-year-old son with SHINE syndrome. I'm also part of the DLG4-SHINE Foundation, a nonprofit run by parents of children with SHINE syndrome. It's my pleasure today to introduce the speaker for today's Science of DLG4-SHINE webinar, Dr. Matt Fuller. Dr. Fuller is the Executive Director of Gene Therapy Research at Ultragenyx, a company focused on developing treatments for rare diseases. He's kindly agreed to talk to us about gene therapy and what it means for DLG4-related synaptopathy. After the talk, I'm going to go ahead and open it to questions, and then we can get those answered. Matt, are you? Oh, there we go. Perfect.
Let's see. Thank you for the introduction. I do want to note that my email address is on here, and if people have questions, they're welcome to reach out to me. Even if I don't know the answer, which is very likely, I can help you find someone that does. One of the really important things about working for Ultragenyx is that they encourage us to use part of our time to help out the community. And so don't hesitate to reach out. What I'm going to talk about today is some sort of intro on gene therapy, but also the considerations for treating SHINE or DLG4 syndrome. So just as a disclaimer, I'm an employee of Ultragenyx, and the opinions that I state here are solely mine, do not necessarily reflect the views of Ultragenyx. So what I like to start with is, what is gene therapy? Because it sounds really fancy, but it's important to understand what's actually going on. And so where I like to start is if you take a cell and you zoom in on it, what you have is you have the nucleus in the middle that contains all of the DNA. And all of that DNA is very compact. In fact, if you take it out of a cell and you stretch it out, it's about 6 foot long. So it's compacted into this structure called a chromosome. When that's unpacked, you start to see strings of DNA, and they're wrapped around these proteins that help compact it. And then if you zoom in a little bit further, you start getting into long stretches of DNA. And those long stretches of DNA are called genes. And importantly, a gene is a basic genetic unit that encodes a function. And if you zoom further into the gene, what you'll see are the actual nucleotides. And so this is the alphabet that you've probably seen on reports that are A, G, T, and C. And so if you take a string of these alphabet and you put them into a function, you have what's called a gene. And a gene encodes something that has an overt function, which is typically a protein, which I'll talk about in a second. And it's important to note that alterations in genes can, but do not always cause dysfunction. In fact, everyone here listening has mutations riddled throughout their genome, but most of them have no known, deleterious effect. And then importantly, as you can see, there's so much DNA in here, right? And all these chromosomes, every gene has an address. And so if this were chromosome 17, what you would have is DLG4 is located on chromosome 17 and P13-1. So the reason that matters is if you're talking to a physician or if you're talking to researchers and they don't know much about your gene, it can be very helpful to give them the gene name and where it is, because that gives them a place to start looking. So DNA encodes for about 20,000 genes, and these genes have discrete cellular functions, and they're utilized by our cells and bodies for our day-to-day existence. And so one of the things that I like to think about is everyone tells you, you need to eat protein and stuff like that. Well, what do those proteins actually do? And so what happens is you have the DNA that we've discussed. It's transcribed into mRNA and it's translated into protein. And what matters is this protein here is what carries out functions. And so these functions, just a couple examples of them, include processing metabolites, contracting your muscles, replicating your DNA in cells, moving other proteins around, activating, inhibiting other proteins, and much, much more. But again, essentially what it is they do all of these functions that are required for our cells to function and for our bodies to function. And what gene therapy seeks to do is replace or repair these specific pathways and molecules when the typical function is not being provided due to the mutation. And so if we go back one slide and you just think about what gene therapy is we have this gene that we know has a certain function. We know it's not functioning as it ought to, and so we're going to replace it in some manner. So to kind of build upon that, a basic definition of gene therapy, this is sort of the scientific definition, is treatment or prevention of a genetic disease via introduction of genetic material expected to provide a necessary function. Or, as I just said, if you know what the gene is that has an issue, you can supply that gene. That gene will now supply the protein that's missing, and that protein can now carry out whatever function it has. There are three main ways that you can approach developing a gene therapy treatment. The first one is add back or addition. And what you do is you add a functional or wild type copy of a mutated or missing gene that's causing the disease. I also like to note that wild type refers to the natural or common version of a specific protein, DNA, virus, or other types of molecules. It's something that you might hear researchers say, and it's not very a common thing to hear. So the most common way that gene therapy is used currently, and the most common way that things have been approved, are addition. So you add back the functional gene. Another way that gene therapy can be done is by inhibiting something. And so this often happens if there is a overexpressed gene or a gene that has a gain of function that's deleterious. And what you do is you can inhibit and activate or knock out, which is remove, a mutated or overexpressing gene that's functioning improperly. This is less common, but there are more examples of this happening. And probably the most in the forefront of our minds, because it's in the news a lot, is editing. And so what you do then is you know exactly what the mutation is. You use these editing molecules called CRISPR, and you change that back to the functional or wild type copy. So while this is the most exciting and it's incredibly promising, it's also the newest. And we'll talk about it in a minute, but it works really well when you remove cells from your body, you treat them and you put them back in the body. We're not quite at a place where we can edit things that are still inside the body, but people are working on that. And I'm happy to chat about that later. So there are three main tools that are used for gene therapy. There are quite a few more that I'm not going to talk about today because they're not used often. But if you do have questions, I'm happy to answer them. So the two most common currently tools that are used, the first is adeno-associated virus. And what that is if you look over here on the right, it is a delivery vehicle. And then if you see here this blue, that's the cargo. That's the gene that is missing. You can also use another virus called a lentivirus. It also is the delivery vehicle, and then the cargo is inside of there. And I'll talk in a minute about why you might choose AAV versus lentivirus, since they are similar in that they're viral vehicles and they contain the cargo. And then again, the gene editing complex is the newest one. It's probably the most complicated. It is the cargo. It's an actual protein, as you can see over here on the right, and it must be coupled with a delivery vehicle. This is something that people are working on quite a bit, and there are some things that are happening clinically, but this is also a relatively new technology. And so, again, it works pretty well when you remove cells from the body. It doesn't work as well currently when you add it into the body. So let's talk a little bit more about the main tools of gene therapy and compare and contrast them. So for AAV, as you see here on the left, it's, although you can't tell from here, it's a pretty small virus. It contains a genome that's 4.7 kb. What that means is there's 4,700 DNA A, G, T, and C. So the alphabet, it contains 4,700 of them. That's the most that you can put in there. And in a second, I'll show you what it would look like if you're making something for a therapy. So the advantages of AAV, it's ideal for in vivo delivery, meaning injecting it directly into a patient or directly into a tissue. It's ideal for gene replacement when you have a gene that's not functioning properly and you want to just add more of it in. It can be a delivery vehicle for CRISPR, but because it's a small genome size, it's kind of difficult to do that. People are working on that. From a safety standpoint, it has very low integration rates. What that means is I'm adding DNA into the cell. Does it actually go into another place of your chromosomes, and could that cause a problem? And that happens very, very infrequently. So it's deemed to be relatively safe by regulatory agencies. There are different shells that you can put around the AAV, and that helps you go to specific cells that you want to go to, whether that be liver or neurons or some other type of cell. And there's extensive academic and industry manufacturing experience. So people have been working on this for the last 40 years or so, And in the last decade, there's been a really strong focus on manufacturing to get the costs of these therapies down. There are a couple challenges, like everything, there are trade-offs. It does have a small genome size. What that means is the gene size that you can put in is limited. If you have a very large gene, such as dystrophin, which causes Duchenne muscular dystrophy, it's much too large to fit in AAV. I'll talk about it in a second, but in the case of DLG4, it does fit into AAV, so it passes that first test. It can be quite expensive to manufacture. It does have a single dose limitation because you're putting a lot of virus into a body. If you're ever interested, we can talk about it. There are things scientifically going on to try to get around this single dose limitation. And safety-wise, there are immune reactions to treatment in small numbers of patients that are not fully understood. We're understanding more and more about them. We're also understanding the max dose that we should give a person. But it's something that really needs to be considered when you're developing a therapy of, how much risk tolerance you have for, potential immune reactions. So for the lentivirus, again, it's a different type of virus. It has a larger genome size. It can take about 9 to 10 kb, which is 9 to 10,000 DNA letters. This is really good for ex vivo delivery, which means you take cells out, you treat them, and you put them back in. And this is often some sort of blood cell, white blood cell, something like that. I'm not going to talk a whole lot about this because I don't really think it's applicable to DLG4, but I did want you to know that it exists in case you speak to physicians or you speak to researchers that want to talk about this. So it exists, but I'm not entirely convinced it's something you're going to be thinking a lot about. So this is what's called a recombinant AAV, or an AAV that you make to treat a specific disease, what it looks like. And so it actually truly does look like this. If you were to put it in a program and fold it, the DNA takes on this shape. And purple on the left and the right sides are something called inverted terminal repeats. Those are viral sequences that are required for making the virus, to replicate it and to stick it in that protein shell. Those are the only viral sequences that are left. Everything else in the middle is stuff that is added to generate this therapy. In the beginning, you need to have a go signal to turn on your transgene. You have multiple choices, and we can talk about those if you're interested. One thing you must have is a promoter, that is the go sequence. And then you can have an enhancer, which can tell you to make the go signal even stronger. In green here, you have the transgene. So in this case, it would be DLG4. That is the actual DNA that you want to go into the cell, turn on, and make the protein to have a function. And then at the end, you have what's called a polyadenylation signal, which is important as it's a stop signal. Just so you're aware, there is an optional sequence that you can put in the middle called an intron. And what that allows you to do is make the genome larger if you need to, but it also usually increases the expression of your transgene, because getting that expression level right is really important, which we'll talk about in a second. But essentially what you need to know is there's a go signal, the gene you need to put in, and there's a stop signal. So what does make a good candidate for addition gene therapy? Again, as I mentioned, there's a small size. So the first question really to me is, does your transgene fit? In the case of DLG4, there are two main variants, which I'm sure you guys are aware of. One is 2,300 base pairs. The other is about 2,200 base pairs. And so what that means is that there is about 2,000 base pairs left to put in these additional sequences that we just talked about. Because it's important to recognize that when I showed you that, and you can see it again here on the right, that it's not just the gene that you need to get in, it's the sequences that tell it to go, that tell it to stop, and tell it to get inside of this protein package. It's important to ask if the delivery vehicle, in this case AAV, can infect and transfer genetic material to the target cells important for correcting disease. And I'll talk about this specifically to DLG4 in a second. But it's important because it's a virus shell. It's really good at getting some places. It's not always great at getting into other places. And so when you're doing your experiments and you're setting up a regulatory package for the FDA, you really want to be able to demonstrate that you're going to target cells that are going to have a therapeutic benefit. And then you also have to ask if the target cells are amenable or conducive to a gene therapy approach and whatever cargo you're trying to put in. And so one example of that is oftentimes diseases of the central nervous system actually will have what's called sort of like a Goldilocks zone where the gene needs to be expressed in between high and in between low. And something high can be called one disease and something low can be called another disease. And so this is something that has to really be worked out with the elements or the, sort of Lego pieces that you put into your cassette to make sure that you get an appropriate amount of expression that doesn't cause further harm. So some considerations for the DLG4 gene therapy, and I know that you already have something going on with a very good investigator, Allison Motry, and I'm sure he's thought of all of these things. But it's good for everyone here to understand as well, because sometimes you need to make tweaks along the way, and understanding these little differences can help you with that. So as I mentioned, the DLG4 coding region is about 2,200 to 2,300 nucleotides. So small enough to fit, with some real estate left for the additional required elements. So it passes that first test. One thing that is important to note, though, is these encode two different versions of the protein called isoforms. And from the little bit of reading that I did, it seems like one is known to be expressed in the brain, and one, I actually don't know the details. It probably is, but it might be expressed elsewhere. So in order to make sure you get a therapeutic effect, you're going to want to make sure that you express the appropriate isoform when you generate this AAV. And that's probably, again, something that's being tested in the lab. So my recommendation is to continue supporting in vitro or cell work and in vivo, which is mouse model or other types of animal model studies, to really fully understand the target cell types and the target expression levels. So all of the data that I could find suggests that excitatory neurons are the main target cell, and those should be able to be targeted by AAV. Now, there are lots of ways to go about doing that we could discuss at another time, but in general, you should be able to reach those cells. However, it's unclear, to me at least, if delivery to other tissues and organs could be necessary. Because if you look in the public databases of gene expression, you see that there is some low-level expression of the PSD95 protein in other tissues at low levels. It's important to do these studies because you could treat only the central nervous system or only the brain and not get full treatment because other cells need it. Or you might find out that the other cells really can do without it or with these very low levels. And so it's important to treat, the CNS mostly. And so those are experiments that you're really going to want to think about over time and will be required for health authority agencies to prove that this is a good treatment to try in people. And as I just mentioned, the target expression levels are really important. And the levels that you'll need for this disease to see therapeutic benefit are unclear. And so you'll want to figure that out because it does inform your vector design. And you'll probably need somewhere between the level of expression of haploinsufficiency, which is too low, and wild type, which is sort of typical expression. You may be able to go above that, you may not be able to. And so you'll need to figure out what the threshold of minimal expression required for therapeutic benefit lies. And also if overexpression would be detrimental. These experiments can be done relatively easily. And sometimes, to be honest, they don't have to be done, meaning if you generate a gene therapy and you see a benefit and you don't see a negative response and you think you're between the haploinsufficiency and wild type levels, you might be good without trying to prove to yourself exactly where those levels are. But if you're unable to do that, then you might need to prove that to yourself. So it's really about understanding the basic biology as much as you can. There are quite a few gene therapy success stories over time. There are 24 approved gene therapy products, 7 are for AAV, and they range for a variety of diseases. So one is for Leber's congenital amaurosis, which is an eye disease. One is for spinal muscular atrophy, which was a muscle disease. Also for hemophilia, which is a blood disease. And also for Duchenne muscular dystrophy, which is a muscle disease. So we've been able to get these to work in multiple parts of the body, and there are many, many trials going on for other target genes and target tissues as well. There also, just for your interest, is one gene editing or CRISPR drug that has recently been approved. And so that field is moving really, really quickly. So when you're thinking about your roadmap, you're thinking about what studies or experiments you want to support. This is really what I sort of think about looking into, the keys to recent successful outcomes. So one of them is having solid basic and clinical science knowledge. And that just has to be determined empirically. You just have to do experiments. But knowing that will help you understand if you're on the right track for the gene therapy you're creating, and if you would really predict that it's going to work in your mouse model, in a non-human primate, and then in a human. High unmet medical need diseases have the most successful outcomes for gene therapy, and it has to do with that risk tolerance sort of equation that you need to do. Having a good cellular tissue target choice. So in the case of you guys, you know that you have excitatory neurons that you're trying to get. You don't think that there are lots and lots of brain cell types that you need to get to. Other diseases don't know that. And so they need to figure that out to really have a successful outcome. And then also this has an increased understanding by health authorities. It was pretty difficult to get these approved, maybe 10 years ago, but it's becoming more routine. There are some limitations. Making these, manufacturing these is costly, it's difficult, but people are understanding this more and more. As I mentioned earlier, there are some safety concerns at time, although those are more and more understood, and I think pose less of an issue as long as people are thoughtful and very careful. And we are continuing to study the immune reactions to gene therapy. So that goes along with the safety concerns, but it can also be something on its own, which is if you have a strong immune response, you might not actually get successful treatment. The person may be okay, but you might not have success. And so they're understanding that more and more as well. And just a couple key takeaways before we have questions. Gene therapy has the potential to halt or treat a disease with a one-time treatment. Now, there are caveats to the one-time treatment, and we can discuss that. But in general, you treat once, and you probably have 5 to 10 years of expression and treatment. And some people have gone longer than that. And there are successful FDA-approved examples of recombinant AAV and lentivirus. But the success is critically dependent upon your solid basic clinical science knowledge, having a targetable cell and tissue choice, and the ability to make the vector. As we sort of discussed, each gene therapy strategy has pros and cons, and you just have to weigh those and test those experimentally before you make final choices. And finally, if you looked very closely at the slide I just showed that had some of the approvals, making these things is expensive, reimbursement is expensive. However, the field is continuing to work on this, costs are coming down, and I think just as important, insurance companies, regulators, and other payers are understanding that this is a different modality that can't be treated as a pill that you just get over the counter. And so they're becoming more amenable to alternative payment strategies. And so that's allowing more and more people to get treated. And with that, I'm happy to take any questions.
Thanks, Matt. That was really, really helpful, especially the targeted information about our protein and our gene. I have a couple questions that I got from an email, and then I'll open it up to others. So the first question is, would this treatment, could it be viable for adults, or do you really have to intervene in the early years?
So that actually goes back to understanding the basic and the clinical science. I personally don't know enough about this disease to understand that, but what I can tell you is there are some diseases, Angelman syndrome is one, and also CDKL5 deficiency disorder, are diseases that are thought to have the ability to actually, trying to think of the right way to put this, essentially halt and sometimes reverse what's happening. But there are some diseases where there are either structural things that happen or maybe developmental things that happen, and you can't reverse. But what you can do is halt. If you halt, then the best thing to do is treat as early as you can. That being said, there are some times where there's still therapeutic benefits seen in an adult population. And I would encourage you to do the experiments to help you understand that as much as you possibly can. I know that's not really an answer, but it really goes back to the disease biology and understanding if there is a window where you really have to treat kids, or if older, people and adults can actually benefit. One other example I'll throw out about that is there are some metabolic diseases that cause the brain to increase in ammonia levels, and it damages those cells and it causes intellectual disability. If you had a disease like that, you really have to treat as soon as you can, because once some of that damage happens, it's irreversible. And so it will come down to understanding exactly how, shine progresses in people.
Oh, thank you. And then the other question was, they were asking about a realistic time frame for potential treatment to be available. And I guess that, after maybe we've done all of our basic science information.
Yeah, so I don't know exactly where Allison is, but let's pretend, he has the vector, which I think he does. He's using AV9.
He
And let's
does.
pretend you have some mouse studies and you have an idea that it's being efficient. You're probably looking at a couple years to do the studies required to file an IND or an investigational new drug application with the FDA. And what that means is you have to demonstrate that you can make the virus. You have to look at the toxicology of that virus to show that it's safe, and you have to show that it is effective. And believe it or not, finding or generating the assays or the tests to show that it's effective can be very, very difficult and can take several years. And so getting that package together, if you're working with people that have done it and know what they're doing, can still take several years. At that point, if it's approved, you will be able to get people treated that are involved in the study. So you can't treat everyone, but you can treat whoever is involved in the study. That probably takes another 3 to 4 years to actually do the study. Now, this depends on the disease, but I'll just say, let's say 2 to 5 years, depending on the disease, to do the study, to get the data back. If you have to make adjustments, that's what's going to make it take longer. If you have to have your dose go up or your dose go down, if you have to change the clinical tests that are helping you understand if it's working. Once you've generated all of that data, then you file a BLA, or can never remember what BLA stands for, but it's for it's a biological license application. So it's application to get an approved drug with the FDA, and then that takes some amount of time. And for that, you also really need to work with someone that's done this before, whether that be a consultant, whether that be a company, whether that be an academic who has done this and been successful. But it's definitely something you want to partner with people on because there's a particular amount of expertise required to do that. So I guess if I were to sort of or summarize that, I would say if you had, a vector right now that you believe in, you're probably 2 to 3 years away from starting a trial, and you're probably 5 or more years away from having something approved. And that's just being honest, that's probably being optimistic. It's really going to depend on your success in the lab, your success in the clinic, and whoever you partner with.
Thank you. Yeah, I think actually that I'm actually kind of excited about that because 2 or 3 years is enough time to get people treated. That's exciting. I'm gonna open it up to anybody else who's if they want to put questions in the chat or if they just want to go ahead and ask a question. We just have a few minutes.
And I'll just add to that while people are opening up that it could take much longer. Let's pretend, that Allison doesn't have luck and there's a lot of optimization that needs to go into the vector. That could take 2 years alone. So I don't want to be, pessimistic. I just want to be really realistic that things have to fall in line. But what's good is people have done this. They have an idea of how to go about it, and you're not, blazing the trail here, which is good in this case.
Yeah, I'm excited that there seems to be a lot of approvals in the last couple of years. So, so that's exciting. So I don't see anyone else raising their hand here. So I think I have one more from an email. I guess this is how do you make the process of rare disease gene therapy treatment financially viable?
I wish I had an answer to that. In fact, I was just recently at a conference with, people that were like CEOs of all of these companies and stuff like that, and they were talking about that. And honestly, they don't have the answer. Part of it is continuing to invest in manufacturing and trying to get that cost down. If you ever watch TV and you see all these crazy pharmaceutical commercials and it's like Skyrizi and all these things and people look happy, most of those are actually called monoclonal antibodies. And that working on that type of drug started in the 80s. And at first it was thought it'll never be viable. It's so expensive. And now we have a bunch of these approved drugs. It took 30, 40 years of investing in manufacturing to get there. But that is one of the things that helped democratize or increase people's ability to get treated with these things. One of the things that's good is we have the benefit of some of that knowledge that we can use. The other thing is we understand um, what that it needs to really be focused on because you know, like, and I'll answer the next question, but like there are a couple of these drugs that I showed that have not been approved in Europe because Europe said no one can afford these, we're not approving them. So I think things like that are also going to put pressure on the pharmaceutical industry to make things more accessible. And I can also answer the question that was in the chat about things being in Europe. That's going to come down to whoever is working on that treatment. Um, it's very common for people to do one of two things. One is either they file first in the United States and then they file in Europe. Sometimes they do the opposite. If the FDA is being, let's say, more complicated than the sponsor is hoping for, sometimes they'll go to Europe and try to do it there. But I think what's important to know is if you start anywhere but Europe, it is a separate license application in Europe. So if you start in the United States, you have to do something separate for Europe, and they'll have different questions. And in addition to that, different European countries will have questions as well. So yes, it can absolutely be available, but it does require specific action on the part of whoever's developing that drug. And one of the best things that you can do to help that is to have your patient advocacy group involved, understand where in those countries these people are that need help, and get that information to the companies. Because if they don't have to look it up and they know, I'm just going to say Finland, is a really important population, then they might focus on that.
Perfect, thank you. I guess we have one more question. How can you draw more interest research funding when the disease is pretty rare? So.
Yeah, that's a really good question that has, all of these have very complicated answers because rare disease is so complicated and unique. So one of the things that's going on now, and I think it really has mixed feelings in the rare disease community, is attempting to find when your disease or gene might be related to something that has a larger indication. And so one example of that is that there are some genes that seem to be implicated in Parkinson's. And so what investigators try to do is sort of tack on what they're doing to Parkinson's research. Because, and the Michael J. Fox Foundation is a good example of that. They have a lot of money and they're willing to cast a pretty broad net and say, we want to see what's involved in Parkinson's. We're fine if you do other things, but this is what we want to know. So that's one way, is trying to relate your disease or your protein of interest to a larger indication. Again, there are people in the rare disease community that aren't really happy with this because they worry that your disease might get lost in the work. And it's definitely possible. So you have to worry about that and be thoughtful of that. The other thing is, unfortunately, you have to push as hard as you can as a patient advocacy group. One of the things that I've always felt was really unfair in rare disease is if you take something like cardiomyopathy, I'm just going to throw out some, term that affects a lot of people. Pharmaceutical companies are willing to look at that because they think there's a lot of money. And that's one of the reasons that they don't want to look at rare disease, just being completely honest, is because there's not a lot of money. If if you're a very large company and you can make billions of dollars on something, or you can make a little bit, you know what they're going to do. And so I see one of the things that patient advocacy groups can really do is fund as much of the basic research as possible to de-risk these programs for these companies. And so if you go to a company and you say, we've done all of this work, we think we have something that might work, they're going to be much more likely to consider it and take it on versus, this horrible disease is affecting people, because they hear that every day, is unfair as that is to say out loud. And so those are the two main things that come to mind. Obviously, the problem is money, both either for the company or for yourself. Honestly, I think we could treat everything if money wasn't enough. Okay, that's a little strong. We could treat many, many diseases if money was not involved, but unfortunately it is right now.
Yeah, it's unfortunate. Well, that's where our role as a patient advocacy group kind of comes in, is try to, mobilize our population.
Exactly. I just want to
Research.
add one other thing. Another thing that we've heard of and seen patient advocacy groups doing is basically using your money as seed money. And so what I mean by that is you don't have to raise $50 million to fund the whole thing. But what you can do, and I think this is what you've done with Dr. Motri, is raise some amount of money allow an investigator to start working on this, and then they can use that data to go get more grants. And so the money that you've given has been force multiplied by helping someone get more money. And I think that's one way to really, I mean, obviously, if you can get $50 million, wonderful. But if you can't, I would argue to not get discouraged and see it as a way to help other investigators get more funding.
Mhm. Yeah, I think that's what definitely what our hopes are for the next stages of the project. So thank you so much. I don't see any more questions in the chat, and I want to be mindful of your time. I thank you so much, and I think we may have further questions kind of coming up now after all the information that you gave us. Very kind of you, and hope you have a happy holiday.
Yeah, happy to help. Don't hesitate to reach out.
Thanks so much, Matt.
All right, bye.